you all for joining us today. I'm Abby, and I'm one of the organizers of this year's Quantitative Seminar series from the Converse Lab. And I want to start today with a land acknowledgement. So the University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. And our lab also acknowledges that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. So today's presenter is Mark Sorel. Mark has been a graduate student at the University of Washington School of Aquatic and Fishery Sciences since 2019, where he is advised by Dr. Sarah Converse of the Washington Cooperative Fish and Wildlife Research Unit. He recently took on a new role as a research scientist at the Washington Department of Fish and Wildlife in the Columbia River Management Unit in January 2022. Congrats, Mark. His professional interests relate to supporting natural resource management using a combination of quantitative modeling and meaningful engagement with managers and stakeholders. In the past, Mark has worked on modeling endangered salmon population dynamics, evaluating the feasibility of reintroducing salmon above high head dams, and estimating sea lion predation on adult salmon. So Mark is going to take questions at the end of the talk today. And just as a reminder, you can either raise your hand and I can unmute you, or you can type your question into the questions poll and I will moderate it to Mark. So thanks everyone for coming, and a big thank you to Mark for sharing your work with us today. And with that, it's my pleasure to turn it over to you.
All right, thank you very much, Abby. Can you hear me? I'm assuming yes, because we were just talking. Yes. All right, thanks everyone so much for being here. Yes, as Abby mentioned, I'm Mark Sorel, and I'm going to be presenting some work today that I've done as part of my PhD dissertation at the University of Washington, where I'm advised by Sarah Converse of the Washington Cooperative Fish and Wildlife Research Unit, and I'm currently a research biologist at the Washington Department of Fish and Wildlife. So the talk today is going to be about an integrated population model that I've developed for Wenatchee River spring Chinook salmon, and we'll just get right into it. And yeah, this is work that I'm doing for my dissertation. It covers, I think, pieces of the first 3 chapters. All right, so I'm quite humbled to be here talking today, given the tremendous number of collaborators and all of the work that's gone into monitoring and modeling salmon in the interior Columbia River Basin. So I want to give a huge thank you to all of my co-authors listed on the left there who have been instrumental in this work, and also to all the agencies listed on the right who have funded the monitoring, funded modeling, carry out the management, and everyone is integral, and I'm just one small part and feel good to be presenting here today on it. So the structure of the talk, it's mostly going to be focused on methods. This is a fairly complicated model, and I'm going to get down in the weeds, but I'll start with a little bit of background and then give an overview of the study system and the whole model, and then we'll get into equations for the process model and the joint likelihood, and then I will finish with some preliminary results and some discussion, and we should have time for questions, and I'd very much like it if I could receive some questions. All right, so starting with population models. Population models are a common tool in both ecology and natural resource management. They can be used to think retrospectively about what governs population dynamics, so things like density dependence or stochasticity, and then they're also often used to understand what a population will do in the future, especially considering a management action or climate change. And so we use them to think about what's population's probability of persistence and how might it respond to a management action, and that might be harvest or some restoration. Population models can be relatively simple in that they can assume that a population is a homogeneous group of animals, but they can also account for population structure, and by that I mean spatial structure, like different parts of a population in different places, or life history diversity, so different animals with different demographic rates and that are behaving differently. So when we talk about salmon, they do tend to have spatial structure and life history diversity. So because salmon spawn in branching stream networks, they tend to have this spatial distribution. And then they do exhibit juvenile life history diversity, where juveniles tend to transition life stages at different ages, rear in different places, and then adults also tend to return at different ages. And so we've got adult life history diversity too. And we want population models for salmon. They're a really valued cultural resource. So for imperiled populations, we want to know what the probability is that they're going to stick around and what the response will be to management actions. And for other populations, we want to know how harvesting them at different rates will meet different objectives. So within the Columbia River Basin, we often we use a mnemonic, the 4 H's, to talk about 4 different common classes of management. Those are hatchery supplementation, habitat restoration, harvest, and the hydro system. And each of those actions affect salmon populations. And in many cases, we're interested in better understanding that. So in addition to caring about salmon, we also monitor them quite closely because we care about them. And so there's a lot of data sets, talking mostly about the interior Columbia, but I guess everywhere. There's a lot of data sets on salmon population trends, demographics. And so, with the combination of wanting population models and having a lot of data sources, our objective here was to come up with a framework that would integrate typical population monitoring data sets to fit an integrated population model for an interior Columbia River salmon population. And we quickly realized that in order to do so, we were going to have to account for spatial structure and life history diversity. So we use the Wenatchee River spring Chinook salmon population as a case study. It's a population that has some really nice long-term data sets that are examples of typical data sets in the interior Columbia. The plot on the right has year on the x-axis and estimates of spawners on the y. We see that the black is the natural origin spawners. They declined through the 90s, at which point they were listed under the ESA as endangered. And at that point, they did make a little recovery, but have been hovering along below the recovery criteria thresholds that are required for them to be delisted. And the red line is hatchery origin spawners, and that's from a hatchery population that is not, it's an integrated population, so it is meant to be genetically connected with the wild population, and it's really used for conservation. There's a different hatchery program that is purely for supplementation, demographically isolated from this wild population, and that's not what I'm showing. And so we see that there's been quite an increase in this conservation hatchery supplementation around the time of listing as well. So in this slide, I'm going to try to do a few different things. I'm going to talk about the life history of these fish. I'm going to talk a little bit about the monitoring, and I'm going to talk a little bit about the modeling. So the Wenatchee River Basin is in central Washington State. It drains a portion of the east side of the Cascade Mountains. As you can see here, the fish spawn in fall in kind of the headwaters of the Wenatchee River, the area that is highlighted in orange. And the majority of the spawning occurs in 3 tributaries: the Chihuahua River, Mason Creek, and the White River. And then fish emerge, juvenile fish emerge from the nests in spring and start to leave, or they emerge in late winter and start to leave in spring. And when they leave, they can be captured in these downstream migrating traps. And one of the interesting features is that these fish leave at different ages. So some fish leave in their first year of life and spend time down here in the main Wenatchee, and other fish stay here entirely until their second spring of life, at which point almost all of the fish in the population go to the ocean in their second spring of life. And so the numbers on these shapes represent occasions in a capture history that we use to estimate survival rates. So some fish are pit tagged, juvenile fish are pit tagged at the screw trap at the mouth of their natal stream, and then they can be recaptured at another screw trap down here. And then when they're going downstream here, we're using detections at this McNary Dam, Bonneville Dam. Then they go out into the ocean. They're in the ocean for between 1 and 3 years predominantly. And then they come back as adults and they can be detected again at these dams going all the way back upstream to Tumwater Dam, which is the last capture occasion that we used, and all the fish have to pass that to get upstream. So that was a big description, but now I'm going to go through the model in more of a diagram. So so start with the spawners. We have natural origin spawners and hatchery origin spawners. So those are fish that were born in a hatchery versus fish that were born in the gravel. And we have information in this plot. I'm also going to talk about the data. So in the data on spawners comes from these red or nest surveys where people are walking the streams. We also have information on the proportion of fish that are hatchery and natural based on recoveries of carcasses. So now I'm going to go into the juvenile data, which comes next. So the juvenile and the juvenile abundance data. The juvenile abundance data comes from these downstream migrant screw traps where you've got fish being captured when they're leaving the natal stream, and then some of those are marked with a tag and re-released upstream, and then based on those recaptures, we get information that can inform capture probabilities in the trap, which can then be used to scale up to total abundance of fish leaving on a given day. The plot on the right has day on the x-axis, and on the y-axis is the average number of fish leaving across all years that we have monitoring data for these 3 natal streams, which I think is between 15 and 21 years across the streams. And the x-axis is actually spanning kind of more than a calendar year. It's more like all of the days that fish from a given brood year could leave. So fish start leaving at very young ages in spring, and then at the same time, you've got fish that are a year old that are leaving, which you can tell the difference of based on their size. And then you have these fish leaving in the summer and fall as sub-yearlings, and these fish will all rear downstream before going to the ocean. So the question was, how do we incorporate this information on daily juvenile abundance into a population model? And what we came up with was to discretize the daily time series into 4 discrete sections, which we'll call juvenile life history strategies. And as you can see, the kind of modality of the average time series supported that, yes, there might be 4 different kind of semi-distinct juvenile life history strategies. So we summed the abundances of daily emigrants within each of these 4 juvenile life history strategies, the sub-yearling emigrants, which I'll sometimes call downstream rearing emigrants because they rear downstream extensively before going to the ocean, and this one yearling emigrant, which I will sometimes refer to as natal reach rearing, because they rear in the natal reach pretty much exclusively until going to the ocean. Okay, and so then we're adding up the abundances of daily emigrants, where you can imagine we'll have 21 annual estimates of summer sub-yearling emigrants, and then that is the way that the data will go into our population model and accounting for the uncertainty. So we get a estimate and really a distribution for each year in each life history strategy. So this is what that looks like in the population model. We've got spawners going to one of these 4, or going to these 4 different juvenile life history strategies. And then, as I mentioned, fish are pit tagged when they're released from this natal stream, some of them, the ones that are large enough. And then we can use the capture histories to inform survival rates downstream until they enter the ocean. And then at that point, we can estimate the the probability of coming back at different ages, and survival back to Tumwater Dam, which is the end of the mark-recapture data that we used. To finish it off, we have adults returning to Tumwater Dam. We take off some fish that are removed to be used in the hatchery brood stock. And then of the fish that are the natural origin fish that are released upstream, some of them will die. And then we also account for the proportion that are female, because this count of reds is really a count of the abundance of females who make the reds. And then for the hatchery origin spawners, we're not explicitly modeling their production at this point, but really they're just coming out of kind of the ether to add into this total abundance of spawners. And then lastly, there is some data on environmental covariates that come in. So we're using tributary discharge covariates in the juvenile production component, air temperature in overwinter, and some marine covariates in small adult return rates. All right, so now I'll just go through the equations that make that population diagram that I just showed go. We'll start with the transition from spawners to juveniles expressing the different juvenile life history strategies. I used a modified Beverton-Holt function for this component of the life cycle. So J here is juveniles from different life history strategies, years, and streams. Alpha is a productivity parameter. Jmax is a capacity parameter, an upper capacity parameter. And then we include this gamma parameter, which is what makes it the modified version. And what that allows for is depensation. And the reason that we included that is because when we first looked at the data on the relationship between spawners and juveniles expressing these life history strategies, it looked like there was some evidence of what accelerating abundances of juveniles with increasing spawners, particularly for some of the younger emigrating life history strategies. And we assume that this might be because of a density-dependent movement where fish are leaving earlier in the presence of higher abundance of densities, higher abundances of spawners. And so including this parameter allowed for the model to fit that kind of behavior. And then we have a log normal error term. And now I will go into how we modeled the log normal process error. So there's a couple of fixed effects. That's the tributary discharge, tributary flow covariates that I was talking about. So thinking that summer streamflow and winter streamflow might affect things like survival and capacity. We included a couple of those covariates. And then we have two random components of this process error. And the first one here was included so that we could have correlations between juvenile life history strategies. And importantly, we wanted to be able to have both positive and negative correlations, because you can imagine there could be environmental things happening that are good for multiple fish that express multiple juvenile life history strategies. But there could also be a dynamic where more fish expressing one juvenile life history strategy means that fewer are available to express a different one, which would lead to negative correlation. So what we came up with was this latent covariate approach, where we've got one omega is a latent covariate of year, and then iota here is a life history and stream specific coefficient or loading for how each of these load onto that annual random effect, which allows for that correlation. And then in addition, we have a component that is just completely idiosyncratic, asynchronous, that is process error that's unique to a given life history strategy in natal stream. So that gets us our abundances of juveniles, and now I'll go into the survival component and all the way back to Tumwater Dam, but we'll start with going to smolt at Bonneville Dam. So we take our juveniles here and we multiply that times survival in 3 different intervals, where T is the index for that interval or the section of the migration. And yeah, that gets us our juvenile abundance at Bonneville. And now I'll talk about how we modeled these survival rates. So yeah, we're modeling survival on the Logit scale. It's indexed again by interval, juvenile life history strategy, natal stream, and year. We have an intercept for the interval, and then we have a set of fixed effects, and those include differences across natal streams or years, and any continuous, or not years, I'm sorry, natal streams or juvenile life history strategies, and then any continuous environmental covariates for a given occasion. And we did group some of the life history strategies in some occasions. So after the first occasion, we assumed that the 3 downstream rearing juvenile life history strategies had the same survival rates because they seem to kind of be behaving the same and sample sizes are a little small. All right, and then we have 2 random components of year for the survival rate. One of those, delta here, is synchronous. So it is a random effect that is affecting all juvenile life history strategies equally. And then we have epsilon, which is asynchronous. It is affecting all the different juvenile life history strategies differently. The reason why we included those 2 is because when we're sampling these random effects of year in simulations, we want to account for the fact that these different juvenile life history strategies are going to be somewhat synchronous, but not perfectly synchronous. And so this allows us to account for that. All right, so once we have juveniles at Bonneville Dam, we multiply. So now we're gonna calculate adults at Bonneville Dam. You can see we're picking up an extra subscript here, which is adult age. So to get the abundance of adults returning to Bonneville Dam from a given juvenile life history strategy, year, natal stream, and age, we take the juveniles at Bonneville, multiply it times the smolt to adult return rate, which is kind of a salmon-specific thing. It's the probability of a fish that enters the ocean in a given year coming back in any subsequent year. And the reason we use that is because we're kind of constrained in estimating distinct annual survival rates because we don't see fish between when they leave and when they come back. And so the probability of returning in a different year and dying in a given year are confounded. So this is a common way to handle that. We have the smolt to adult return rate, and then this psi, which is the conditional probability of returning at a given age for fish that return. So if you return, what's the probability of returning at different ages? And for us, that's 3, 4, or 5. The smolt to adult return rates were modeled basically the same linear predictor as the downstream survival rates. And here I'll show the conditional probabilities of returning at different ages. It's modeled in multinomial logit space where alpha is an intercept for each age, but one of the ages, age 4, is fixed at 0. We have a fixed effect, and that is the effect of being a juvenile life history strategy where all of the 3 downstream rearing juvenile life history strategies are combined. So we're just allowing different return age probabilities for downstream rearing and natal reach rearing juvenile life history strategies. And then just a synchronous random effect here. It didn't seem like we had enough power for an asynchronous random effect of year. And the synchronous random effect of year is distributed bivariate normally, where we're accounting for correlation between proportions of fish returning at different ages, because if more fish are returning at age 3, a higher proportion, we would expect there might be fewer returning at age 5. All right, and then to get adults back up to Bonneville Dam, back up to Tumwater Dam, we take the adults at Bonneville Dam and multiply them times survival in 2 intervals going back upstream. So, and this upstream survival is modeled pretty similarly to downstream survival and smolt to adult return rate, with the exception that we are assuming that juvenile life history strategies and natal streams all have the same upstream survival, but we're allowing survival to vary by adult age at this point, and we're only including a synchronous random effect of year. All right, the last part is to get adults from Tumwater Dam to spawning and bring in the hatchery fish. So to get the abundance of natural origin female spawners here, we're summing the adult returns to Tumwater Dam over juvenile life history strategy and adult age, and subtracting off fish that are removed for broodstock, natural origin fish that are removed for broodstock, We're then multiplying that times the proportion of fish that are female in a given year in natal stream, and times the survival rate from Tumwater Dam to spawning. And the proportion of fish that are female is a random effective year. To get the other component of spawners, the hatchery origin spawners, I mentioned we're not explicitly modeling their production. Instead, we're estimating the proportion of hatchery origin spawners, and then based on the proportion of hatchery origin spawners and the abundance of natural origin spawners, we can calculate the abundance of hatchery origin spawners. The proportion of hatchery origin spawners was a random effective year, which was mostly to regulate it away from 0 or 1 in years where there wasn't much data. But when we're going to be simulating this prospectively, this is usually always going to be a management, something that's under management control. And then for the total, to get the total abundance of spawners, we add together the hatchery and natural, and this is what then goes into the juvenile production function. So that is the completion of the population process model. Last thing I'll say about the population process model is that we initialized it with estimates of natural origin spawner abundance and the proportion of hatchery origin spawners in the first 5 years, where we couldn't calculate natural origin spawners based on the process model yet. All right, now I will go into the joint likelihood, or the observation model for this, which is how we use data to estimate these parameters in the process model. So on this slide, I'll show how the data on the abundance of juveniles and spawners came into the model. We'll start with juveniles on the top. So here, M star hat is the log mean of juveniles from a given life history, natal stream and year, from that model where we summed across days. And sigma M star is the log standard deviation of that estimate from that observation model. And so the log mean is normally distributed around the log of the latent juvenile abundance, with the observation error based on the estimate of uncertainty from the screw trap model. Moving down to spawners, X obs is the count of reds from each natal stream in each year, and the log of that is normally distributed around the log of the latent spawner abundance in a given stream and year, with this observation error. And this is an estimated parameter, but with an informative prior based on previous work evaluating the observation error in red surveys. All right, so that was, okay, so yeah, the capture history data, the likelihood for the capture histories it's a multi-state model, because we have survival, and then we have that probability of returning at different ages, and we're also accounting for imperfect detection, so we're estimating detection probabilities or recapture probabilities. And so we're calculating those likelihoods using the forward algorithm. The information on the proportion of females, as I understood it, the best information came from observations at Tumwater Dam, and I didn't have information on the number of fish that were examined, so I just had this PF obs, which is the proportion of females observed at Tumwater Dam. So somewhat arbitrarily, I chose to model this as normal in logit space with an observation error of 0.25. Where PF is the latent proportion of females. For the pre-spawn survival rate, survival from Tumwater to the spawning, again, didn't have data here to inform annual rates, so we assume a constant rate across years and streams, and this is informed by literature values and an informative prior that is drawn from developed literature values. All right, and then the proportion of hatchery origin spawners, as I understood it, there's some extra binomial variance in the observations here. So C here is the the abundance of carcasses recovered, and CH is the abundance of those that are of hatchery origin. And because there was some extra binomial variance in these recovery probabilities of the hatchery origin carcasses based on environmental features and things, I chose to just model this as Poisson, which has more variance, observation variance than a binomial, where the expected value is the product of the number of carcasses and p-Haas for the number of hatchery carcasses. So that's it for the observation model. I developed this thing in template model builder and was using maximum marginal likelihood, but then now I'm drawing posterior samples with TMB Stan, and those are the results I'll show today. And I want to mention that I use penalized complexity priors judiciously throughout this whole model. So penalized complexity prior is a prior that puts more weight on some simpler model, like a coefficient being 0. And with all of the coefficients that I included, for example, different effects of juvenile life history strategy and natal stream on all the demographic rates, the variance of the model would have exploded, and there's no way it ever would have converged or given anything sensible if I didn't use these penalized complexity priors. And I use them in quite a few different places, and they were really, really handy for smoothing this thing. All right, so now I'm just going to show a few preliminary results. Here we have the latent spawner abundance on the x-axis and emigrant abundance on the y-axis. We've got natal streams in the columns and juvenile life history strategies in the rows, where the later emigrating juvenile life history strategies are lower down. And the points represent posterior medians with 95% credible intervals. The red line is the expected number of emigrants for a given number of given number of adult spawners, and the red shaded envelope is a 95% prediction interval. So what we saw here that was interesting is that the younger emigrating juvenile life history strategies seem to exhibit less negative density dependence, and you see more negative density dependence as you go down, suggesting that as you get more spawners, you're going to actually have a higher proportion of fish leaving the natal stream at a younger age. Here we have ocean entry year on the x-axis and smolt to adult return rate on the y. And we've got two panels, one for each group of juvenile life history strategies. I think what's interesting here is if you squint, you can see that there is quite a bit of synchrony between these, but they are not perfectly synchronous. So we've accounted for that in our model. And I think on average across the year, downstream rearing juvenile life history strategies had slightly higher smolt to adult return rates. And one reason for that is because the downstream rearing life history strategies also tended to return at a slightly younger age. So here we've got ocean entry year on the x-axis, the proportion of fish returning at different ages. This would be a posterior median on the y, and the colors represent ages. You can see a slightly higher proportion of age 3 fish amongst downstream rearing juvenile life history strategies, and a slightly higher proportion of age 5 lower proportion of age 5 amongst downstream rearing juvenile life history strategies. This is kind of my favorite plot here. We've got spawn year on the x-axis and natural origin returns on the y, and the different colors represent juvenile life history strategies, and these are posterior medians. What's cool about this to me is that getting this information required integrating the juvenile abundance data and the demographic survival rate information. So, and what we saw here is that in Mason Creek, most of the adult production is of fish that had exhibited a downstream rearing juvenile life history strategy, those sub-yearling emigrants, whereas in the Chihuahua, it was more of a 50-50, and in the White River, the majority of the adults had exhibited a natal reach rearing juvenile life history strategy. And then lastly, this is proportion of hatchery origin spawners. This is another thing that we're tracking in the model, and a quantity that is of quite a bit of management importance, because it has a lot to do with genetic introgression between the wild and the hatchery strains. And so, you know, it's been variable historically, but this is something that the model tracks. It's important. All right, so in conclusion here, the model, you know, we estimated the production of these different juvenile life history strategies, and we saw, based on the density dependent functions, that it appears that as we get more spawners, we're going to have a higher proportion of fish rearing downstream. And you know, overall, I think it's quite important to be able to understand where fish in the population are rearing seasonally. And one of the reasons for this is because when one of the management actions is habitat restoration, and that habitat restoration happens in a given place. So it could be in a downstream rearing habitat, or it could be in a natal stream. And so knowing what proportion of the population will be in a given place and kind of benefit from a given habitat restoration strategy could be really valuable. And then also thinking about climate change, it could really help us understand the impacts of that, which will presumably be heterogeneous across the Wenatchee River Basin. Today, I talked a lot about the fitting of the model and the retrospective analysis, but one of the main reasons we developed this is for prospective modeling. And for this endangered population, we want to know what's the probability of extinction and what will the abundance be in the future, and especially under alternative recovery actions. And we've been working with policymakers in the Wenatchee River Basin to identify management strategies that they're interested in better understanding the outcomes of. And then I'm currently working on simulating those with the model, and that's what's going to be in my 4th chapter. All right, and then just getting back to the big picture, I think that we're at a time where I see population structure being incorporated more and more in population models. I think there's a lot of new tools that are coming out which make that easier to do, things like data integration, spatial modeling, and I think that's a really exciting thing, because it's going to help us make predictions about the impacts of management actions and climate change, which are themselves often structured spatially or affecting fish with different juvenile life history strategies in different ways. And with that, thank you very much for your attention. And as I mentioned, I would be very happy to entertain some questions. Thank you.
Thanks so much, Mark. That was awesome. I've got a couple, but it looks like there's already a couple questions in the chat. So this one is from Trevor Branch. And Trevor says, what is the form of the penalized complexity priors, and how is that added to the model?
Yeah, thanks. That's a great question. Thanks, Trevor. Okay, yeah, I did not get into the details of that, but I'll do my best to explain it. So the form of the penalized complexity priors are, so one place where we're using them are some fixed effects in the survival rates and the demographic rates. So on those betas for like either a effect of an environmental covariate or a categorical effect of, say, juvenile life history strategy. And so the form of the prior, and it's from that paper that I highlighted by Daniel Simpson, I think from 2019 or maybe it was 2016. The form of the prior is an exponential prior on, no, I'm sorry, it's a normal prior on the coefficient with an exponential prior on the standard deviation of that normal distribution. And as I understand it, that particular penalized complexity prior is equivalent to ridge regression. So it's really just a form of ridge regression, essentially. But yeah, they were super handy. I included that kind of prior on a lot of different covariates. And then I also included it on like random effective year standard deviations, when those random effective year were kind of a little squirrely to fit. Slapping an exponential prior on the standard deviation was quite helpful.
Awesome, thanks, Mark. And yeah, I just want to remind folks, if they want to elaborate on their questions, feel free to use the raise hand feature and I can unmute you selectively. I will ask you one, and this maybe will reveal my naivete about salmon. But what do you think? It was interesting to me that there's so much variation in life history strategies across those 3 natal streams. And in particular, the I think it was the one that starts with an N. Sorry, I forget the name. But what do you think is driving that variation and diversity?
Good question. Well, I think that there's probably a lot of things, and I'm sure I won't think of them all off the cuff, but one of them, I think the 2 primary ones would be density dependence and bet hedging. So there's all this habitat, you know, the best spawning habitat might be in the natal stream high up, but that doesn't necessarily mean that the best rearing habitat is there. So, you know, and it's interesting actually, one of the cool things about salmon is like, there's a whole nother stock of Chinook salmon that spawn in lower down in the Wenatchee River, and they exhibit a juvenile fishery strategy where they leave the natal stream and go to the ocean as sub-yearlings. So that potentially opens up habitat down there, but the bottom line is that there's habitat downstream that can be used. And so if there's a limitation on how many fish can be supported in these natal streams, life history diversity could be a releasing of that bottleneck. The other thing is that it's a bet hedge. So if there was I don't know, a landslide or I don't know, some bad thing that happened in a natal stream or a downstream area, if you've got fish in a different place, they still have a chance. And you know, there's some interesting things where fish that exhibit, well, I don't know if I want to get into this, but fish that exhibit downstream rearing juvenile life history strategies and some other study areas have been shown to be more likely to stray to other natal streams. So it could also play a role in kind of like spreading of the population or main connection, but it's definitely an interesting phenomenon.
Yeah, definitely. Yeah, and I think your model reveals some really interesting things about that. So cool. Before we get too far from Trevor's question, Devin Johnson would just like to add to Mark's prior penalized complexity prior description. It functions like the penalty in a GAM and shrinks coefficients to 0 if they're not necessary for model fit.
Yeah, thanks, Devin. Feel free to unmute Devin. Devin is the person who turned me on to the penalized complexity priors a few years ago, and I'm very thankful for that. And I am glad that he brought that up. The connection to a GAM is really a good one. It's an ability, it's a smoother, you know, in effect. And I think that's one of these tools that I'm talking about that's allowing us to account for individual heterogeneity in population models, is the fact that, you know, you don't have to estimate the survival rate of one life history strategy and the other life history strategy completely separately. You can using these tools, these smoothers, you can say what is the support for a difference, and in the absence of that, we'll just pool that information, which is really helpful, and you see it in spatial modeling too.
Cool, thanks, Devin. I have another question for you, and it's one that I've been thinking a lot about in my own work, and that is one of the other things that I think was cool about your talk is the sort of the synchrony between the populations that was revealed. And I'm always curious about, like, to what extent is that an artifact of your model structure, and to what extent is that kind of emergent as a result? And it's not a fully formed thought, but I would just wonder if you can riff with me.
Yeah, no, that's good. Yeah, it's so I hope it's not an artifact of the model structure, but Yeah, so I guess, right, it's this kind of variance decomposition thing where, you include a random effective year that is common to all juvenile life history strategies and a random effective year which is asynchronous, and then I hope that the statistics kind of sort out that variance, and that they're trying to kind of reduce those random effects as tight as possible, and kind of partition that variance into how much of it can go in this synchronous chunk and how much of it needs to go in this asynchronous chunk. Yeah, I think it's an interesting question. That's kind of my philosophy on it, not being that well trained in statistics.
Yeah, sure. I don't have an answer either, really. I guess one thing that comes to mind too is just that you have so much more data from one of those streams than your other two as well. So to share some structure makes sense there.
Yeah, no, that's interesting. Right, the different amounts of information.
Got another question for you from Brielle Thompson, who says, do you think any additional data would help improve your model estimates? You've got mark-recapture and survey data, but what else? Sorry for the dings in the background.
Thanks, Brielle. Good question. And I feel like I thought about this question because I was wondering if someone may ask me this. And the first thing that popped in my head was kind of a smart ass answer, which is that it feels like there's so much data actually that exists that I'm hesitant to try to think about more data that I want, because I haven't incorporated tons of data sets that already exist. For example, like, there's pit tagged hatchery origin fish that I could bring in that could potentially inform some of those demographic rates. There's other populations in the Columbia River which are, have similar monitoring data and could potentially inform, they share some demographic rates. There's some information that can be shared there. And there's other data sets that I haven't even thought of. There's fish that are getting pit tagged in the natal stream, not in the screw traps, but just by electrofishing. That's certainly one area that I think would be cool to expand to, is if you could get some estimates of survival in the natal stream, you could start to think more explicitly about those trade-offs between staying in the natal stream and leaving, and asynchrony in survival rates between the natal stream and the downstream area, which I can't really get at when I'm only using the data from fish that leave and downstream. So I guess maybe I twisted your question around a little bit, but I do appreciate having the opportunity to talk about some of the other data sets that are available that I think could be brought into this framework, or this framework could connect with.
Yeah, nice. Your answer sort of reminded me of the quote that you had up there about the penalized complexity priors, like that they must be used judiciously, and data integration is the, is the same. We've got another question from Stacy Ambergy, who says, how do you expect this model to be explicitly put into practice by managers moving forward?
Yeah, that's a great question. Something that I'm starting to think about more and more now. So when we met with managers, we mostly assembled a group of people who were working on habitat restoration in the basin and hatchery supplementation. And there are management decisions being made about both. So for the hatchery, there are a few different dials that I think are quite interesting. So one is the size of your hatchery program, which is like how many broodstock you have, how many juveniles you produce, and therefore how many adults you get back. But then when they get adults back, they're often taking some of those adults and what they're calling surplusing, which is just not needing them for brood stock, but not wanting to release them onto the spawning grounds because they don't, you don't want to just flood the spawning grounds with hatchery origin fish that can have a really bad genetic effect. So there's a management control about how many fish you produce, but then also about how many fish you take into the broodstock and how many fish you release upstream of the Tumwater Dam. And that's really related, what the right number of fish to release is really related to the capacity of the natal stream, the capacity of the whole system. And so I think it's going to be really interesting to explore kind of those dials and see what the optimal, see where there's trade-offs and what the outcomes are of different levels of supplementation there. The habitat restoration question, I think one of the interesting pieces is about whether the effort is put in the downstream areas or the natal streams, and that could actually interact with the hatchery supplementation piece because of that phenomenon that we saw where the more spawners you get, it seems like there's going to be more fish going down at a younger age and rearing in that downstream habitat. So I think that there's some interesting questions there that I hope we can get at. And thanks for that question, Stacy.
Yeah, nice. Thanks for the answer, Mark. Just, you mentioned earlier in your answer to another question too, that there's another population that has, different life history and rears downstream. Is it kind of like a two birds, one stone approach with the habitat restoration downstream?
Yeah, I'd never thought of that, but probably. I don't know much about that. It's a summer Chinook salmon population, so it's not spring Chinook. I've never thought about that, but yeah, absolutely, right? Yeah, I think that if you do restoration in downstream areas, it could benefit, right, other populations that are also using that habitat. It's a really good thought.
Cool.
And of course, like this is a very salmon-centric model, but yeah, there's benefits to habitat restoration, I suppose, that are outside of just spring Chinook. I guess there's steelhead in some of these populations, and I mean, I even admit that there's other species other than salmon that exist. Yeah, amphibians, birds, it's crazy.
Awesome, thanks, Mark. I got one more question for you if you've got time.
Yeah, oh, absolutely, thank you.
Cool. It's from Brielle again, who wants to know about modeling latent movement. So like, it seems like the way that you've approached it is kind of to do this discrete movement between certain spots, i.e. the dams. And have you thought about, or do you know of other work that treats modeling movement differently? Like continuous space or something like that?
Yeah, that's a really good question. I mean, my mind immediately goes to, there are models that have been developed for looking at survival and fish behavior in the Columbia River hydro system, which kind of work on like more of a daily time step. And I believe they use advection diffusion equations, which I don't personally understand myself, but to model kind of the spread of fish and the downstream migration. And so they're looking at, and the data that they're being fit to is daily arrivals at Bonneville Dam. And when you get into management things at the dams where you're like spilling water on a given day and things like that, then that information on, within season dynamics really matters. So I'm definitely not an expert in that, but if you're interested, sometime I could dig up probably some papers on that advection diffusion approach to modeling that downstream migration.
Brielle says she's interested.
All right, Brielle, we're on.
Cool. Well, thank you so much, Mark. I'm not seeing any other questions trickle in, but if I'm missing you, please raise your hand. Not seeing anyone. Just want to give you a round of applause.
Yeah, thank you. It's an honor to present a quantitative seminar. I've been a fan of the seminar for a long time, and so this is quite an honor and a lot of fun. So thank
Yeah,
you.
well, thanks so much for coming, and thanks everybody. We'll see you next week for Stacy Ambergy, who's going to talk to us about
Yes,
this. So should be a good
gonna
one.
be good. That's gonna be really good. Definitely
Yeah.
gonna be tuning into that.
All
All
right.
right. Take care, everybody. Thanks, Abby.
Bye.